This is the third video of the ionotropic receptors first lecture. In this lecture, we will talk about ionotropic receptors. We will introduce what type of ionotropic receptors exist in eukaryotic cells and which is their subunit composition. So, ionotropic receptors are also called ligand-gated ion channels, and are membrane-bound receptor proteins that respond to ligand binding by opening an ion channel or pore and allowing ions to flow into the cell. And they can have both excitatory and inhibitory functions. Most of ionotropic receptors are heteromeric proteins with multiple types of subunits that group together to form an ion channel. Ligand-gated channels can be divided in three major families. There is the ATP P2X receptors, also known as purinergic receptors. The second family is the CYP loop receptors, and the third family is the glutamate-gated channels. So for the first family, the purinergic receptors, these are composed of three subunits. Each subunit is composed of a protein with two transmembrane domains, and both N-terminal and C-terminal domains are facing the intracellular space. For the second family, the cis loop receptors, they are pentameric receptors, meaning that they're composed by 5 different subunits. Each subunit has 4 different transmembrane domains, and both N-terminal and C-terminal are facing the extracellular space. And the last family, the third glutamate-gated channels family, are tetrameric receptors composed of 4 different subunits, in which each subunit has 3 different transmembrane domains and also an internal loop that faces in the membrane. And for this case, the N-terminal is facing the extracellular space, while the C-terminal is facing the intracellular space. So there are different ligand-gated channels we can find in eukaryotic cells. And here there is a list of how between the different 3 families we can distribute the different major ionotropic receptors. So for the the cis loop receptors, they include the nicotinic acetylcholine receptors, GABA and glycine, and also serotonin receptors. For the family that corresponds to glutamate receptors, we have AMPA, NMDA, and kinase receptors. And then for the purinergic receptors, we have the P2X or P2Z receptors. And here there is an example of one of each of those receptors. For the first one is the purinergic, and this is a representation of what we've seen in the previous slide. This is how acetylcholine, GABA, or glycine receptors look like for each subunit, and this is how the glutamate receptors look like for each subunit. So different receptors have different subunits, as we said, but there is variability within the subunits that they can occupy for each receptor. So for the glutamate receptors, as we see, there are tetrameric receptors with 4 subunits. In the case of AMPA receptors, there are 4 types of subunits that can combine differently to form different kinds of AMPA receptors. That means that they could have one GLUR, 2 GLUR2s, another GLUR3, and different combinations of those 4 subunits. In the case of NMDA receptors, we have 5 different subunits that can combine differently to form the tetramer. And the same applies to the kinase. There is discrepancies in how the kinase receptor subunits are called. Here I put the most recent nomenclature that they include, that they call these subunits from GLUC1 to GLUC5. But they also receive other names depending on which source you look at. Then for the cis loop receptors, the ones that are formed by 5 subunits, they also have a variety of different subunits. They can compose each of the receptors. In the case of the GABA receptors, there are subunits corresponding to alpha, beta, gamma, delta, epsilon, and p, and there are different isoforms of each of these subunits. And similar things happen for the glycine receptor, the nicotinic acetylcholine receptors that we will see in more detail in the next video, and the serotonin. And the same applies to the purines receptors. And the most important thing here is that you need to know that different subunit composition provides different properties to a receptor for the same neurotransmitter. So, what kind of signals do ionotropic receptors produce? Well, as I mentioned earlier, they can be excitatory or inhibitory. And when a neurotransmitter binds the receptor, it triggers a conformational change that allows ions to flow selectively through the channel. So the subsequent current flow sets up a potential difference called a postsynaptic potential, which may be excitatory or inhibitory depending on the ion selectivity and the membrane potential. So excitatory neurotransmitters like glutamate or acetylcholine results in the intracellular accumulation of sodium ions and a decrease in chloride and potassium ions, depolarizing the cells and generating an excitatory postsynaptic current. On the other side, inhibitory neurotransmitters like GABA or glycine results in intracellular increase of chloride ions and hyperpolarization, generating an inhibitory postsynaptic current. And we will see more about the electrophysiological properties of the different ionotropic receptors in way more detail in the last lecture of this session of ionotropic receptors. In this example, I want to emphasize the diversity of the different receptors and their different subunits composition and how subunit composition determines the properties of the receptor. This is an example of 2 different glutamate receptors composed by different subunits that are represented as different colors. And below you can see where the responses of the 2 different glutamate receptors when they're exposed to the same agonist. And the response is completely different despite the fact that they both respond to glutamate and they're both glutamate receptors. In one case, the agonist produces a fast inactivity while the other receptor causes a steady state current. So these are 2 completely different responses mediated by just having different subunits composition. This is just an example to represent how diverse these subunits can be expressed, not only at different time points during development and adulthood during the life of an animal, but also region specificity. So here the different channels as well as the different functions have different effects, and this is determined by when and how these different subunits are expressed. And in this particular case, this is an example of NMDA glutamate receptors in which different subunits are expressed differently through aging, and this provides a great diversity in the amount of responses that these and other ionotropic receptors can mediate. And here I leave you with a short quiz that reviews some of the most important concepts that we've seen in this introductory video.